All right, I think it's ready to go. It's 11:07. That's our usual start time. Say goodbye to the Little Mermaid and Sebastian the Crab. Okay, good morning, everyone. We're here to talk about vaccines against viruses. I chose 3. This is the one you care about the most, the coronavirus, but it's the one we know the least about. So I put it last because of that. This is the greatest success story in the history of vaccination. Smallpox, influenza is the flu, and that's a different story. So let's go ahead and start. You'll have chances to ask questions. I think I put in three places where you can ask questions, so please be thoughtful as you see the data I'm presenting. So I'm starting with smallpox. None of you have seen a smallpox victim, but this is what they looked like when they existed on this planet. So these are the pox, these little bumps, and this is a person who got covered with the pox, then it scabs, and this person did recover. So if it didn't kill you, you were immune for life. And this has been known since ancient times. But people didn't have any way to treat it or any way to prevent it from occurring to you until about 1800. So I get to show you that data. So the virus, most viruses have about the same structure. There's some nucleic acid material in the middle. In this case, the smallpox virus has DNA in the middle. And then on the outside, there's some sort of envelope or membrane, and it has proteins on it. So these little things that look like red trees, those are proteins that are responsible for allowing the virus to bind to cells and enter those cells. So I said on the first slide, smallpox was a deadly disease. And this is historical data from London. In the 18th century, that's this period right here in the middle, 1 in every 13 deaths was caused by smallpox. Majority were children. The vaccine appears right here at the end of the 17th century, right around 1800. So you can see there's an effect here in London. My next slide shows similar idea from Boston. Deaths from smallpox are these red bars. You know, thousands and thousands of people are dying of smallpox. And then the vaccine comes to Boston in 1800. And after that time, you know, the deaths drop to less than hundreds, right? Less than hundreds. In this case, less than 1 because it's per 100,000. So maybe 1 person died of smallpox in Boston in this time period. So this is showing the effect of the disease statistically. Sorry, the effect of the vaccine statistically. This is another story. Smallpox was brought to the New World. It did not exist in the Americas until the Europeans brought it. And in Mexico, the effect was devastating, and it's been documented. You can see this is all written in Spanish. I thought that was appropriate that we get the discussion of what happened in Mexico in Spanish. So if you can read Spanish, you can read this. Viruela is smallpox, an epidemic of smallpox, probably introduced by Hernán Cortés in 1519. The population at that time in Mexico, these are the Aztecs. The Aztecs, close to 22 million, and smallpox killed 1/3 of them. Very quickly killed 1/3 of them. And we have these historical pictures from that time period showing these are Aztecs covered with pox, dying of this disease. So that's a devastating epidemic. 1/3 of the population dead. So, to the rescue comes this English physician, Edward Jenner. 1796 is the first vaccination. So this is a famous painting, and he's scratching the boy's arm here and introducing something that will protect the boy for the rest of his life from smallpox. And what he used in that year, 1796, is something called the cowpox virus. So this is the smallpox virus. This is the one that killed millions and millions of humans. This is the cowpox virus, which is very similar. They're closely related viruses, genetically, structurally, and that's what allowed this to work. This is a picture of cowpox. It infects cows. So Edward Jenner used the cowpox virus, and that's what he is putting into this boy's arm. He didn't understand how it worked, but he quickly collected data showing it does work to protect humans from smallpox. So being infected by a related virus gave humans immunity against the smallpox virus. And historically, he thought about this because he noticed that milkmaids, those who were milking cows, were very unlikely to get smallpox. So what in their occupation made them immune? He thought maybe it's this, maybe it's this. So it was a brilliant leap of scientific intuition that gave us the ability to fight smallpox. So took about 200 years. The first vaccination is 1796. Took about 200 years, but by 1980, smallpox is gone from this world. So you can see it here in the graph that there is no smallpox after 1980. So that's all due to the vaccine. Well, I shouldn't say all. It's mostly due to the vaccine. In the 20th century, most of these cases were in India. India was one of the last countries to get common use of the vaccine. So there were hundreds of thousands of cases in that part of the world, but there was a concerted effort, World Health Organization, let's vaccinate every human, and then we won't have this disease anymore. So, as I said, the greatest success story in the history of vaccination. We don't have smallpox anymore. No one in the future will die of this disease that killed millions of people in the past. The way the vaccine works, and Edward Jenner had no idea of this, but we do now. With modern science, we know how it works. It's all about antibodies. So antibodies are your defense against viruses and bacteria. So a vaccine causes your body to make these antibodies. And an antibody is a protein. Some of you may have seen this slide before. It's a protein. It binds to a specific target. And it's made by cells in your immune system. So I'll try to explain how all that works. This is a 3D model of an antibody. This is basically a 1D model, one-dimensional model of an antibody. These are amino acids. So it's made of amino acids. We had that in yesterday's talk when Adam Farshid was talking about making his hydrogels out of amino acids. These are natural proteins made of amino acids. Okay, antibodies can neutralize virus infectivity. I'm going to show a 45-second YouTube video. To understand it, you need to recognize this shape here is a virus. This white surface below is a human cell. The viruses are infecting the cell. Antibodies will come to the rescue. For this, I have to exit out of my presentation. Let's see what happens. Here we go. Skip the ad. And 3, 2, 1, bing. All right, so these are blood cells streaming through your blood. The white ones are the ones that can make antibodies. Here's the virus. It's going to attack a human cell by binding to it. So that's really important. That allows the virus to attach to the cell. Often there are many viruses and they can all attach to the cell. And here come the antibodies. So these shapes here are the antibodies, and you'll see them intercept the virus. Intercept the virus. Before the virus can bind to the cell, the antibodies will get the virus. And there they go. This virus cannot infect the cell and is instead going to be gobbled up by a different kind of white blood cell. That's called a macrophage. Gulp, it's gone. Virus is cleared. You don't get sick if you have antibodies. Oh, it came right back. All right, cool. So that's the idea. Antibodies are what you need. The vaccine helps you make antibodies. This is a really important graph, so look at it carefully. The y-axis is antibodies made. Those are the proteins that will neutralize the virus, get rid of the virus. You can think of kill the virus. Antibodies do that. And the title of the slide is Primary and Secondary Antibody Responses. First time the virus attacks your body, your immune system doesn't know, what is this thing? Where did it come from? What should I do? So there is essentially a lag before your body starts making antibodies. So in this case, it starts making antibodies after about a week. So this is the time you're getting sick. You're saying, I don't feel so good. Virus is multiplying, infecting more and more cells, and then your body starts making the antibodies, and then you say, I start feeling better. Oh, I'm doing okay now, because the antibodies are killing the virus. So you get this wave of antibodies produced, and it makes you get better. It heals you. Virus is probably cleared by this time, so the antibodies are not needed anymore. And nature, being what it is, says, if we don't need antibodies, let's not make them. It's wasteful. Let's not make them. So you go and say, okay. But the key thing is, if you're seeing this virus for a second time, then the antibody response is much faster, almost immediate, and a bigger response. So if you have a response like this, you don't get sick at all. Before the virus can kill enough cells to make you sick, your antibodies are already killing the virus. So this is the whole basis of vaccination. On the first slide, I said, if you didn't die from smallpox, you are immune for life. This is, I'm infected with smallpox. I made enough antibodies, I survived. If I see smallpox again, no problem. I'm immune. I have antibodies against it. All right, so it's specific cells that make the antibodies. I thought this was worth showing you this little YouTube video here. So these are called B cells. They're a type of white blood cell. They are specific for certain things. So in this cartoon, it's a bacterium. And these are the antibodies that protect you from the bacterium. Could be a virus here, same idea. So I hope this will be meaningful. These cells are going to multiply. That's what cells do. And this particular cell, let's say it recognizes smallpox. And if you have enough of them, you'll be immune to smallpox. So there's a cell dividing.
Plasma cells.
I think I'm going to do this without sound. And this one is therefore going to start shooting out antibodies that'll clear the virus. And these are called plasma cells. And there they are, shooting out antibodies. Go find the virus. Go intercept the virus before it can infect a human cell. So these are the ones that make the antibodies that were seen in the graph previously, but they don't live that long. They live about 2 weeks. Whereas this upstairs here, this is a different kind of cell. It doesn't shoot out antibodies. You see, it has these kind of antibodies on the surface, but it doesn't shoot them out. And this one can live forever. This one can last your lifetime. And it's called a memory cell. And it says, we remember. We remember that smallpox infection from when you were 5 years old. We will defend you if it comes again. So that idea is the basis of vaccination. So B cells make antibodies. A single B cell can replicate, duplicate itself, produce many, they're called daughter cells, that all produce the same antibodies. And some of these daughters can survive, they're called memory cells, for a human lifetime. So these make the antibodies that clear the infection right now. These memory cells remain in your body for the rest of your life, saying, if needed, we can make a bunch of these warrior cells very quickly. We know what to make, we know how to make it, we'll do that. All right, so this is the last slide of this section. This is a powerful image. This is from Dr. Jenner's house in England. It's a museum now, and he was the one who first inoculated or vaccinated people against smallpox. It's from 1901. Both boys were exposed to smallpox. The boy on the right, who looks perfectly healthy, had been vaccinated. The boy on the left was not. So this boy, his immune system was saying, we've seen that virus before, no problem, it's taken care of. This boy, this is the first time his body is seeing smallpox. His immune system can't fight the virus effectively. He's covered with pox. That's the history of smallpox. We don't have it anymore. All right, this is my pause for questions. So I'll look at the chat and please give me some questions about what you've seen so far. Oops, sorry.
I have a question, Mr. Sogo.
Yeah, let me try to get this to work. Yes, good. Go ahead.
It's my understanding that after an immune response, you have antibodies in your body. It never goes back down to zero once you have a specific antibody. They're always kind of circulating, and then that's how your body knows to mount a defense if you get the infection again. Is that correct?
It's not really the circulating antibodies that are important.
Mm.
Because this can go almost to zero here.
Okay.
If you look at the scale,
Right.
This is 1.
Right.
This is
Right.
100. So it's not a linear scale, it's a log scale. So it's not antibodies you have, it's the
memory cells.
Memory cells, which you start. One of the things that I saw says you start with maybe 1 million different B cells, and they're all specifics to something.
Mm-hmm.
But you only maybe have one for smallpox in the beginning. But after you've been exposed once, maybe you have 1,000. And so that's what these are.
So antibody testing to see if you've had a virus, is that something you can only do after a specific time of having that virus?
That's a really good question. That's an excellent question. I don't know the answer exactly. Presumably, it does matter where you're testing. If you're antibody testing a year later, maybe the antibody titer is so low that you can't detect it. Okay, let me look at chat and see if I've got anything. All right, I've got. Okay, Taro is saying he recommends a show. So John Adams, that's the right time period. John Adams is the 2nd president of the United States. So right around the time when the first vaccination is done. Thank you, Taro. Okay, good. So he's saying in that show, they're talking about the first vaccinations, which if it's John Adams, they're probably occurring in Boston. All right, Morgan Zevnik says, why does the smallpox virus have DNA, not RNA? That's really important for what I'm going to talk about next. Some viruses are DNA, some viruses are RNA. It does matter quite a bit. Luke is saying, can memory cells be extracted and injected into other people? Transplanting cells is really tricky because, let's see if I can do it. Let me use this slide. You have your body, your immune system will know when foreign cells are injected. So if we'll use Luca Alganian as example. If some of Luca's cells got injected into my bloodstream, my body would say, foreign cells, destroy, 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 destroy. So that is good because that's how the whole system works. These bacteria are foreign cells, destroy, 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 destroy. But it makes it so that you can't easily transplant cells from one human to another. It causes the cells you transplant die. This is a real big problem with like kidney transplants. If you're saying, I need a new kidney, can I take one from this donor? The answer is usually no. It will be rejected. It will be destroyed by your immune system. So that's why they have to do careful matching and saying, is this a close enough match? Thank you for the question, Luca. El Gurley. Yeah, she's saying, aren't there a lot of symptoms we get from sickness caused by our immune system's reaction? That's also true, absolutely. Sometimes, and with this disease that we're fighting right now, the COVID disease, much of the damage is caused by an overactive immune response, where I was just saying, attack, attack, destroy, destroy. That can sometimes be targeted to your own healthy tissues. Attack, attack, destroy, destroy. Diabetes type 1 is caused by that. Your body's immune system attacks part of you and says foreign cells, attack, attack, destroy, destroy, and you lose the ability to make insulin because the pancreatic cells that make it are destroyed by your own immune system. Yeah, so those are immune system, sometimes called autoimmune disorders, immune system malfunctions. They do occur. They're significant. They're important. All those can happen. Okay, thank you for the questions. They were excellent questions. So this is smallpox, great. Make a vaccine, cure the disease. It's gone from the world. No one will ever get smallpox again. Influenza, the flu, different story, different story. Flu shots, I'm hoping some of you have gotten a flu shot. And they're vaccines. They're an attempt to say, see this virus now so that your body can make antibodies, and when it comes back in a month or 2 months, you'll say, no problem, we have seen this before, we know how to kill it. So that's what the flu shot is for, is to give you that first glimpse saying, you may see this later. Usually you get your flu shot like in November. Say, we'll give you this shot in November so that in February, when you see the virus, when it comes to infect you, you're prepared. Your immune system is prepared. Okay, so once again, there's a picture here. All viruses look roughly the same. This is the nucleic acid inside. In this case, it is RNA, not DNA. And on the surface, there are these proteins. The blue ones are the most important ones because they allow the virus to bind to the cells, and that's what triggers the infection. But unlike smallpox, you can get the flu every year. And that's because the virus mutates. So here is a headline from 2017 saying that a mutation in a particular protein caused the flu vaccine to be not very effective. This is from University of Pennsylvania School of Medicine. So here I'm showing you the same picture as before. This is a zoomed in view of one of these blue spikes. They're called hemagglutinin, hard word to say, usually abbreviated HA. And it's this H here is hemagglutinin. But the blue regions here are regions that mutated, so it looks a little different. Here's another slide getting a little more detail. These are all pictures of the same blue protein on the previous slide, the hemagglutinin. And now they're saying, well, at this position here, there was a change in the amino acids from N, which is glutamine, to K, which is lysine. And here's isoleucine to valine change. Here's a glycine to it's glutamic acid, glycine to glutamic acid change. So these changes occur naturally. And because of that, they look a little different. And the whole basis of the vaccination is your body has to say, I've seen that before. And now, oftentimes, these mutations make it so your immune system says, I don't recognize that one. So it's a disguise, right? It's a disguise. The flu virus is constantly changing its surface, putting on little disguise masks saying, how about this mask? How about this mask? How about this mask? And those are caused by mutations in the RNA. So because of that, the virus changes every year. And so you have to be prepared against not just one virus, but all the different variants of the flu virus. That makes it a lot harder to make a vaccine. All right, there was an earlier question, I think it was from Morgan, talking about RNA versus DNA. RNA viruses tend to be much less accurate in copying their RNA. So the smallpox virus was really, really accurate at copying its DNA, making sure the next virus, its offspring, are exactly the same as the first one, exactly the same as the parent. So he said, I am an excellent virus. I will make my progeny just like me. And for thousands of years, really, in the history of humanity, that was true. The smallpox virus was able to spread from human to human and replicate and kill people and replicate and kill people. So make it just like me. I'm perfect. And the flu virus is exactly the opposite. Yeah, let's just make something that's kind of like me, but it can easily be different. And if I make 1 million offspring, then let's survival of the fittest. Survival of the fittest. Maybe one of my offspring that's mutant will do better than I did. So that's the nature of this RNA virus. Much higher mutation rate. Much more difficult to make a vaccine. You cannot make a flu vaccine that says once you've had this, you're immune for life. That cannot be done. So instead, CDC, Center for Disease Control, has to predict what to put in the vaccine for that year. So they put 4 flu viruses in the vaccine, 3 or 4, and they're trying to say, oh, this is probably the right one to use this year. And they do it with this test, which is pretty. So I'm going to try to show it to you and hope you understand it. If you understand nothing else, you can just say, it looks pretty. So these previous season vaccine virus, they have lots and lots of viruses at CDC, flu viruses, different strains, and they say, well, is the virus that's circulating out there now similar to last year's? And so they do this test with red blood cells. My next slides will explain how it's done. But you can see it's pretty easy to tell whether something happened, because the picture this is visible with the naked eye. You don't need a microscope, you just look at it. And you can see that this virus reacts about the same as A. B is similar to A, but C is quite different. So what CDC tries to do is say, if C is circulating now, we need to put something like C in the flu vaccine for this year. Virus B here, we probably don't need to vaccinate it against that because it's similar enough to what you saw last year that you're still immune to virus B, but we need to give you immunity to virus C. All right, so I'm hoping that picture is somewhat meaningful. It's complicated. But this usually happens in February. CDC is deciding in February for what goes in the vaccine that we're going to give in November. Okay, so here's, I think I get, this is about 2 minutes, and it's going to show how do you do that test. How do you do this test with red blood cells? Here's how it's done. Oh, I better turn up my volume. Hold on a second, because this one you do have to hear them. Settings. All right, co-host will tell me if it's not quite the right volume.
Hemagglutination inhibition is a very convenient way of measuring the amount of specific antibody against a virus in serum samples, and it can be done quickly and easily in the laboratory. And it relies on the fact that certain viruses are able to clump red blood cells, as we saw in the hemagglutination assay. As you'll remember from that assay, red cells on their own will fall to the bottom of a tube or a well and produce a button that you can see with the naked eye. But if you mix them first with the right sort of virus, they will clump the virus and prevent it from forming a button at the bottom, but it will form a shield instead. The hemagglutination inhibition assay relies on specific antibody, preventing that shield formation. And this works as follows. The specific antibodies bind to envelope glycoproteins on the surface of the virus, and thereby prevent those envelope glycoproteins from binding to the receptor molecules on the surface of the red blood cells. And so the virus does not cross-link the red blood cells and doesn't form a lattice at the bottom of the tube. But the red cells now are able to fall freely to the bottom of the tube and form a button. So we can turn this into a convenient assay of antibody concentration in a very similar way to the hemagglutination assay you saw before. So we start the same way with a series of round-bottom wells containing equal volumes of saline. We then make serial dilutions of the serum samples instead of the virus preparation. The serum samples are serially diluted in the saline solution, after which we add a constant amount of virus, typically 4 hemagglutinating units of virus, and then we add the red blood cell suspension and allow the components to mix and the red cells to settle to the bottom of the tube. At the end, we note the last well in which a button has formed, and we say that's the end point of the titration. And by looking at the dilution of serum that went into that well, we can describe the titer of the original serum in terms of its ability to neutralize that particular antibody.
Okay, so that shows how the test is done, and ACR students will recognize those tools that were used. So let me go back here. And so you can see the visual results. You just wait half an hour. You have to do a lot of pipetting, put the right things in the wells, but then you just wait half an hour and you say, okay, virus C is not very much like virus A, so we need to put in this year's formulation something that reacts with virus C. So that's a guessing game. It's a guessing game. Sometimes they guess wrong. And the virus, the vaccine isn't very effective that year. Usually it ranges in effectiveness from about 20, maybe 15%, if it's a really bad year, 15% effectiveness to 60-something%. All right, so then this next video is going to show how do you actually make the vaccine. It is a huge production effort because they need to make millions of doses. And if we get a vaccine against the coronavirus, again, we'll need billions of doses. So that's a real challenge. But this is a nice video that shows how is the flu vaccine made. Every year it is made. Okay.
A vaccine is a very simple means to train your body and to prevent it from being infected by a bacterium or a virus. We are manufacturing here on this site influenza vaccines, seasonal and pandemic influenza vaccine. We get these eggs from about 14 different farms, spread all over Germany and right into the Netherlands as well. The manufacturing process of our influenza vaccine starts with the inoculation of the influenza virus into the eggs, followed by an incubation at well-controlled temperatures under well-controlled conditions. During that incubation phase, the influenza virus is multiplying. In the second stage now, we harvest the allantoic fluid from the eggs. This allantoic fluid now contains millions and millions of copies of influenza virus. In the next stage, we purify the virus from the allantoic fluid, and we also split the virus into small particles and inactivate it chemically in order to prevent it from being able to further replicate. In the fourth stage, we sterile filter to yield a sterile suspension of purified influenza split virus with each of the 4 influenza strains we received from the WHO reference laboratories in order to formulate a quadrivalent vaccine containing 2 B strains and 2 different A strains as a base composition for this vaccine. The manufacture of our bulk antigen takes us about 30 days, followed by another 30 days necessary for the formulation, filling, packaging of the vaccine. Our whole manufacturing process and the success of what we are doing depends on the people we have. This year we will produce approximately 50 million doses of influenza vaccine, delivering a very safe product which is able to protect people from serious infections.
Okay, so that was from Germany, but it's done in many countries, and it is mostly done by private companies. Now, it's not a government thing. It's, you know, the private companies are making the vaccine. They're making a profit on it, but it's a public health venture. All right, so he mentioned the inactivation. There's some chemistry in that. These are molecules that can be used to inactivate a live virus. So this is the original live virus here. You don't want to inject that into a human because they'll get the flu. So instead, you take the virus and you treat it chemically so that usually it's bits and pieces. He mentioned that it's bits and pieces. This can't replicate, can't make new viruses, but it can allow your immune cells to say, "Ooh, we've seen that blue shape before." So when a real virus comes, your immune system says, "There's that blue shape. We saw it before. We saw it in October or November." When the virus comes in February, your immune system has memory cells that will make the antibodies and bind to that. And so you don't get sick. All right, so that is a different story than smallpox. Smallpox, 1 vaccination, good for life. Flu, 1 vaccination per year helps protect. It's not 100% effective. Okay, so pause for questions here. Okay, first question is from Luca Elganian. And his question is really interesting. It's saying, if the immune system is directed by the nervous system, could psychoactive drugs interfere with the body's instinct to destroy foreign cells and tissue. All right, as far as I know, the immune system is not controlled by any nervous system inputs. So the creation of the antibodies and the proliferation of cells is sort of the immune system talks to its other cells. So there's this huge network of communication between immune cells, but it's not controlled by nervous system. So it's sort of a self-contained virus fighting pathway. And that's perhaps why, you know, Al Gurley's question about does it sometimes get out of control? Yeah, absolutely. There is no conscious control. There's no nervous system control. So sometimes the communication between cells cause them to be too destructive. Waiting for another question?
I have one question.
Please.
So when we were looking at the mutation of the flu virus, and they were saying that sometimes you can have a vaccine that will work against one that you have. Does the mutation have to be large enough to change the structure of the protein in order to make it and make your immune system have to make new antibodies?
It's difficult to say. So that's why if you were to say which changes will make it so this antibody no longer recognizes that is difficult to predict. Though with computer technology, it's becoming better. So the idea that maybe in 10 years we can really be good at predicting that may be true. But that's why this assay is done, because you just see how well does it work, but there's still a lot of guessing here, saying I think this is going to work, and it's not understood on a molecular basis. So I think your question is saying, can we get to the point of saying, yeah, if this mutation occurs, this is what we should do to make a good vaccine, and we're not at that stage. Humanity is not at that stage yet.
Right. From my understanding, like this year they delayed, or last year they delayed the flu vaccination because they got information from the southern hemisphere that went through the flu season first, and then reformulated the whole flu vaccination, which is why it came out late this year. So it's that's part of the guessing game, and that's part of how long do we wait, right?
Because this process with the from choosing the vaccine viruses, saying these are the 4 we're going to use, to the production of the 50 million doses, that's about 6 months.
Oh wow.
So it's not trivial. It's not, and there are many scientists thinking about how do we speed up that process. 6 months is too slow to do it well. So how can we speed that up? So instead of using the eggs, there are other ways of growing the virus, or influenza anyway, and that may help. And recombinant DNA may also help. If all you need are the bits and pieces, you don't need to start with the whole virus. Can we just make the bits and pieces to start with?
Could you just make the protein, so to speak?
Yeah, exactly.
Got it.
So that's future science. Let's go forward to my last section. Yeah, I can do it well. All right, so this is the one you care about, the coronavirus. Some of you may think there was no coronavirus before this one, but no, there's a family of viruses. They've been with humans for thousands of years, and this one just happens to be one that kills people much more effectively than any of the others in the past. So the same structure as you've seen before, some nucleic acid on the inside. It is RNA for the coronavirus, and then these proteins on the outside that allow it to bind to cells and infect them. Good news first. Some scientists are studying antibodies raised against viruses and saying how well do they work. So unlike the flu virus, where you have the really convenient, easy put red blood cells in a dish, see it in 30 minutes. This is much harder to do. You have to actually measure, did the virus get in? So this is a paper from March 2020. And it's interesting because, see, this is not the first coronavirus that killed people. This is the third, really, it's the third. But there was in 2003 an outbreak of a disease very similar to this one. It was caused by coronavirus. It was severe acute respiratory syndrome. And so people studied that virus, and this experiment is done with antibodies that were raised against the first virus. So do antibodies against the first virus protect against the second virus? Really important for a vaccine. And their findings were that, yes, antibodies against the first virus protect against the virus we have right now. SARS-CoV-2 is the virus we have right now. But it's not perfect. So this is infectivity, virus entry into human cells. Here's the virus. Does it get into the cells? That's the assay they're doing. If you have antibodies, it won't get in. These blue things are antibodies. It won't get in. So here, antibodies against the first coronavirus, basically 100%. Perfect. Virus can't infect the cell. This is now the second coronavirus, CoV-2. And you can see it knocks it down by about 90%. So the antibodies are effective at preventing the virus from getting in, but it's not 100% effective. So that was the first data that I learned about with antibodies and the coronavirus, and it's encouraging. This is the question that many scientists are trying to figure out. This virus has only been with us for about 6 months, and so the data is not complete. But from what, this was published April 6th, so a month ago, the data from this study was coronavirus seems to mutate much slower than the flu virus. That's good news. That's good news. So it's not changing its surface proteins as fast as flu does. So that suggests maybe you can make a vaccine and your body will recognize the coronavirus. I mean, ideally for life, that seems unrealistic in this case, but at least for a year, 2 years, 5 years. So then, you know, this was done by a computer scientist at UCSD. That got cut off here. It says University of California, San Diego. And looking at all the data that he could get based on the current data, it seems as though this SARS-CoV-2 mutates about 25 mutations per year. Seasonal flu is 50 mutations per year. So that's good in a way and bad in another way. Smallpox had much fewer than this, and that's why it's gone. This is a newer publication, May 5th. This was kind of frightening to me. But they're trying to do the same thing that was on the previous slide. Count mutations. How fast are mutations occurring in this virus? And at the time of this paper, May 5th, to date, we have identified 13 mutations in spike that are accumulating. So this is, you don't need to understand everything here, but in the middle is Wuhan. That's where the virus originated. So there was some ancestral virus. All these branches came from that one virus. All of these branches are mutations from the one virus. So you can see there are lots of different coronavirus mutants that have been sequenced, is how you do it. You look at the DNA sequence, you say, is this the same as the original? If not, how different is it? So many, many, many mutants have appeared. This one talks about a specific one. And I'll show you the graph about why. Well, actually, I'll read you the text first, then I'll show you the graph. The mutation, spike D614G, is of urgent concern. It began spreading in Europe in early February, and when introduced to new regions, it rapidly becomes the dominant form. The title of the paper is Mutation Pipeline Reveals the Emergence of a More Transmissible Form of this Virus. So let me show you the picture, and then we'll talk about it briefly. So this is a map of the world. Dates. Before March 1, this is the month of March. And this is by country. So I'll start with the map. So the yellow is the Wuhan virus, the original. D is the amino acid, it's aspartic acid. G is glycine. So the original Wuhan virus had aspartic acid in position 614. The mutant has glycine there. So you can see here in Asia, it's almost all yellow. That's all the ancestral form of the virus. In the United States, it's almost all yellow. So the early viruses that came to the United States came from China, Zoop. It's the yellow virus. Now, as we look at what happens here in a very short period of time, the blue virus is taking over. So this is global here now. And you can see, let's take France, for example. France started with mostly yellow virus, but by the time March came around, it's all blue virus. So when scientists see something like this, they're saying, oh, the blue virus must have an advantage over the yellow virus, survival of the fittest. This blue virus is more fit to survive and spread in humans than was the original yellow virus. So this is bad news, because if this is a more easily spread form, then more people are going to get sick. So you probably care most about United States. Doom, you can see the blue form is spreading. There have been some speculation, not proven yet as far as I know, that New York was so hard hit, and California, it's like, what? And there's been some speculation that in California, we still have the yellow form, and New York got the blue form. And that's why millions of people in New York got infected, whereas California is still seeing thousands. So if the blue virus spreads across the country, which it almost certainly will, then that may be bad news for us because of this mutation causing easier transmissibility. Okay. All right, that's the end of my talk. So once again, I'll give you a chance to ask questions. I'm on schedule, 12 o'clock. Oh, I should have put my Jeopardy song here, huh? Calms me down.
Mr. Stogo, I have a question.
Uh-huh.
Is there any indication that if you have antibodies for the yellow form, that you are not immune to the blue form?
Excellent question. I haven't seen any data on that. It is probable, based on previous data from previous viruses. It's probable that if you're immune to yellow, you're at least partially protected from blue. It's really, really unlikely that this one mutation will make it so that you have no immunity. But
Cool. Also, just as a side note, I had the antibody test done this morning, so I'll find out tomorrow.
How interesting. Okay,
Yeah.
Was it the little cassette, or do you know?
I was getting some routine blood work, and my doctor just threw it in there, so it was just a blood test.
Okay, so they took your blood, and they'll let you know sometime soon.
Sometime soon, today or tomorrow, he said.
Okay, excellent. Yeah, that will be really interesting. Okay, I am not seeing any questions in the chat at this time, so I will say that's the end of my talk. Thank you for being here, and hope to see you again next week. So bye-bye for now.
Thank you.
Thank you.
Thank you.
Thank you, Mr. Sogo.
Thank you.